it's meant to be an informal talk. So please feel free to ask questions, anytime during the talk, or if I get, too much in some technical detail or something is not clear, please feel free to interrupt me right away and ask questions. And also after the talk, I allowed some time, and please feel free to ask questions about my work or in general about autism. I'm not going to talk specifically about treatments or other clinical aspects. I'm not a physician. I have a PhD, so this talk is specifically intended to give you an idea about the state of mostly preclinical research in this field. So I'm going to share my screen now. May take a couple of seconds to load. All right. Can you see my screen? Can you see my presentation? Okay, great. So yes, probably you all know some of these introductory concepts, but I wanted to just start by mentioning that autism spectrum disorder is a group of conditions that are characterized by difficulties in social interactions primarily and communications, along with restrictive, repetitive behaviors, interests, and activities. It's been estimated somewhere around 1 to 68, but depending on the diagnostic criteria of the way it's measured, it could be even higher, like 1 in 50 children. It's been very clear that genetics plays a major contribution to the cause of autism, but it doesn't account for 100% of the causation. And I'm going to talk a little bit about this later. And what is also becoming clear is that these genetic alterations affect brain development primarily, neurodevelopment. And but environmental factors also have an important role, but they have been much more difficult to study. So we have less clear information about them, but we know that the immune system plays a role, and often environmental and genetic factors work together to lead to the expression of the disease. So I don't want to get in detail about this, but I would like to stress that it's been pretty thoroughly investigated and it's been demonstrated that vaccines do not cause autism. But intense behavioral and educational therapies like ABA, for example, especially when started early in life, can improve significantly the social and communication skills of kids, and then later on help them find jobs and live more independent lives. Pharmacological treatments, currently there's no one treatment or drug for autism, but pharmacological treatments, drugs have been used to manage specific symptoms and comorbidities of autism. And I'm going to talk a little bit about this too. And as I mentioned, unfortunately, there is no cure for autism yet, but understanding the biology and at a fundamental level and modeling it in animals or cells is the way to get to these treatments. And that's our hope, that by understanding what's going on during neurodevelopment, how these genes affect neurodevelopment, is going to help us directly find the treatments. So you probably also know, I'm sure you know, that autism is a spectrum disorder. So it's very heterogeneous. It includes, a number of previously separate conditions, but some of these definitions are actually quite broad. So, for example, pervasive developmental disorder, not otherwise specified. It's a fairly broad definition, and some people, people with autism have are very highly functioning, but others have more severe impairments. So it's also a spectrum of functional impairments. Also, autism, as I will mention, is very heterogeneous when it comes to causes. So many genes contribute to the risk of autism, and these genes are very, very different. But often autism is a cause of other neurodevelopmental disorders, such as Fragile X syndrome, Phelan McDermid syndrome, or many copy number variations, tuberous sclerosis. But with many of these mutations, not all kids who have the same mutation or the same disease have autism, and the autism is not equally severe in them. So we have a fairly complicated problem, because we have to try to find what is common and what is the cause in a very diverse type of condition that affects almost each person in an individual way. So another aspect that is important considering is that autism is often so-called comorbid or associated with other conditions. So it's not just the core aspects of autism are social deficits, language impairment, and repetitive behavior, but many kids with autism have intellectual disability. About 30, up to 30% by some estimates have seizures, some form of seizure disorders or epilepsy. Other types, sleep disorders, sleep problems are very difficult for parents. Mood disorders, anxiety, attention deficit and hyperactivity disorder, obsessive compulsive behaviors, and also other non-nervous system dysfunction like GI, gastrointestinal disorders, and immune dysfunction. So sometimes these other conditions are actually more difficult to deal with for, like, everyday functioning than the core autism symptoms. So a lot of parents, seizures are extremely scary and could be fatal. Sleep disorders, mood disorders, aggression could be very difficult to deal with. So it's also very important to understand how these comorbid disorders relate to the core symptoms. So I mentioned earlier that genes play a very important role in autism, and it's by some estimates 50 to 90% of the cause of autism is genetic. But that genetic doesn't necessarily mean it's inherited from parents, and I'm going to talk a little bit about that. But what is very understanding the genetics of autism has been really a huge breakthrough toward discovering new treatments, because once we have genes, we know what pathways in the brain are affected. We know how to model these genetic mutations in mice or in stem cells. So we have something to study. Otherwise, autism, when we would just look at the clinical features, would be a very heterogeneous and very, very difficult condition to address, to target therapeutically. So coming to genes, there are different types of, one thinks about a genetic mutation, or even more like a gene causing autism. So in fact, genes control bodily functions, and when they don't function properly, often because of a mutation, they could lead to disease. But these so-called mutations, or even more recently, they're called variations, because not all of these genetic changes are immediately causing disease. Some of them may just increase risk for disease by a small fraction. So it's interesting to know that there are two general classes of these genetic variations, or we could call them mutations, but for simplicity. Some are rare, but are highly penetrant, which means that they have a very strong effect. So some of these genetic forms, genetic mutations, cause syndromic forms of autism, like mutations in the gene called SHANK3 or mutations in the gene called CNTNAP2 cause a developmental disorder syndrome that is highly associated with autism. But in other cases, these variations could have a very small effect, and they could be also present in parents, or it could be present in people with no no sign of autism or no other deficit. But then, so they are actually quite common in the population, and many of us carry some of these mutations. But then, when they sort of co-aggregate in one person, then they can tip over the balance toward the clinical condition. Or some of these common variations, common genetic alterations, could also combine with some environmental factors, like an immune response or something of immune challenge or something of that sort, that could again tip the balance over autism. So a little bit more about these mutations. Again, there are several ways to classify them, but a little bit earlier I mentioned that genetic doesn't mean inherited. And that's because many of these mutations are so-called de novo. So they actually occur in the germ cells of the parents. So often in the sperm cells of parents. So the parents do not carry the mutation, but the mutation occurs spontaneously in the egg or in the sperm, and then the child will have that mutation. And all cells of the child will have the mutation. So many of these high effect size, rare genetic mutations are these de novo type. And then other inherited mutations could be inherited, but could be often recessive in parents. So basically, they don't show up in the parent, even though the parent has one copy. But when two of these mutations come from different parents, then the disease may be expressed. Or some of them are these common mutations, common variations that I just mentioned, which have very small effect size. And all of us have a few of these. But when they aggregate somehow in one person, they may sort of tip a balance and lead to autism. So that's sort of that's a slightly out of date cartoon, but we know that out of all the genetic cases of autism, about 3% are these inherited single gene mutations, like for example Fragile X. 5% are some kind of chromosomal alterations, chromosomal breaks or anomalies, which I'm going to talk a little bit about. And then 5 or 10% are these de novo, so-called de novo, these new mutations that occur in germ cells. And about 80 or 85%, we know that they are of genetic nature, but we don't know what causes them. We don't know the gene, we don't know the mechanism. So there's a lot more to find out about that. But basically, even about what we know now, we can already learn a lot about the etiology and pathophysiology of autism. So these individual mutations that I mentioned, some of these de novo mutations, are spread across all chromosomes. And when the mutation, these occur spontaneously, and sometimes they occur in between genes or in regions of the DNA that are not so important, so we don't notice them. But sometimes when they occur in specific genes, and especially in genes that control the development of the brain, then they cause disruption of these genes and could lead to autism. Similar conditions, similar type of mechanisms occur in, for example, genetic epilepsy disorders or genetic, disorders of other systems in the body. So specifically the genes that control neurodevelopment, when they are mutated, in many cases, they could lead to autism. But this type of knowledge is actually could be useful scientifically and clinically, because we can study these individual mutations. And even though these are rare at the level of the population, like let's say one specific mutation that I'm pointing here may be present in 5 kids worldwide. But as I will discuss, we can make that mutation in mice and study those mice and develop drugs using those mice. Or we can take stem cells from these patients and use them to develop personalized treatments for those patients. So even though these type of mutations are rare, they could be very useful scientifically, but also clinically. So these are just some of the genes that are affected by such mutations, and you may, if you had genetic testing, you may recognize some of these. So another class of mutations are these larger chromosomal deletions or duplications. So in this case, a big portion of a chromosome is removed or is duplicated. And one well-known example of this is Down syndrome or trisomy 21, where an entire chromosome 21 is triplicated. So instead of us having the person having 2 copies, they have 3 copies. But in some of these cases, it's actually only a small region of the chromosome. So one chromosome is normal. Each person has 2 chromosomes, 2 sets of chromosomes, one set coming from the mother, one set coming from the father. And they all have the same set of genes. So in a way, it's a protection mechanism. If you have a mutation on one of the chromosomes in one gene, you still have that gene from the other parent. So it could still be functional, at least, half functional. So it somehow it could compensate for the disease. But in some cases, this deletion of or duplication of a chromosomal region could still lead to increased risk of disease. And I'm going to talk about one of these a little bit later. So in this context, I'm going to talk a little bit about genetic testing and in what ways it could be useful. So
yeah, I'm going to be downstairs.
So genetic testing is not routinely done in autism, but it's done more commonly in, for example, pediatric epilepsy syndromes. And many of those cases also are comorbid with autism. So often, kids who have autism, if they have epilepsy, they may be tested for genetic mutations because of that. But knowing that genetic mutation actually could help in several ways. So it could alter the course of treatment. In some cases, kids with a specific gene mutation are sensitive to some drugs versus others, or they have some side effects versus others. They need special forms of care. Sometimes these kids are prone to some medical conditions, like seizures, which may occur later, or obesity, or kidney problems, or for example, kids with SYNGAP don't feel pain, so they are prone to injure themselves because they don't have this version of harmful experiences. This can help families make family planning decisions. Some of these tests actually are provided in microarrays, which is a faster and cheaper way of testing. But some of these more in-depth methods are becoming also more financially accessible. So for example, whole genome sequencing would cost about $1,000. So that is becoming more within reach of families. But it's very important to combine this with a genetic counselor, because a lot of these mutations need to be interpreted, and it's not a very clear-cut response whether that mutation is the one that causes the disease or not. And these type of mutations could be useful now or in the future for enrollment in specific clinical trials or repurposing specific drugs. So, for example, some anti-epileptic drugs could work in some kids that have autism and seizures with a specific mutations, but other drugs may not work. So it could help clinicians prioritize which drugs to test first. And last but not least, this knowing what genetic mutation the child has could help parents organize or join these advocacy groups and support groups like the ones that I listed here, but there are many of them. And as these kids have specific needs depending on the specific genetic condition and specific parents can share experiences, parents can share the name of specialists, families can participate in clinical trials, families can advocate for the specific condition for funding or regulatory policy changes. So understanding the specific mutation could help in many ways today and also in the future. So a little bit more about the science now, and please feel free to stop me and ask me questions if I get too technical here. But one of the conclusions from these many genetic studies that have been done is that many of the genes, even though they are very different, and here genes are represented by these circles, and the lines are sort of connections between the functions of these genes. Many of these genes encode either proteins that regulate the expression of other genes, so they control these developmental genetic programs, or regulate synapses, which are the connections between brain cells. So this already tells us a lot about the causes of autism, is that these genetic mutations somehow happen in genes that control brain development or control synaptic connection formation, and somehow disrupt these processes. So as I mentioned, many of these genes affect synapses, and synapses are the sites where brain cells communicate with each other. So this is a cartoon of a synapse, and here you have one brain cell sends a signal to another brain cell, and these brain cells form networks like in a computer, like a microchip in a computer, and that's how we process information, and that's how we control our movement. That's how we control social behavior or speech. So if there are some problems in this, in these synaptic processes, those physiological functions are not done properly. And then gene expression, like during the development, these proteins that are concentrated in synapses, and this is a cartoon view of all the autism risk genes and the proteins they encode and as they localize to synapses. They are made in the nucleus and then shipped out to the dendrite. So many of these genes that are mutated in patients with autism affect these processes. So what happens to the brain? And there have been, again, using mostly these mouse models in which you can take a gene that it's been mutated in patients with autism, and the clinical presentation of those patients, and you try to replicate that in a mouse. And I'm going to talk a little bit about that more. But then with a mouse, you can do a lot more studies on brain function, and you can understand much more about what's going on between the genetic mutation and the clinical outcomes that you would see in patients. So in the brain, there are two types of brain cells. Some are called excitatory, and they excite the other brain cells, and some are called inhibitory. So it's almost like on-off switches, or almost like little transistors that have the capability of switching on and off the current. So these type of brain cells connect with each other, and they control each other's function. So in the brain, the number of these excitatory cells and synapses and inhibitory synapses needs to be balanced, because if there's too much inhibition, our brain quiets down, and it's almost like being on an anesthetic. If it's too much excitation, it can lead to seizures. So the brain has to be very, very controlled when it comes to the balance between excitation and inhibition. And many studies in animals, animal models in mice, but also studies of in patients, for example, the high comorbidity of epilepsy with autism has led to the idea that in patients with autism, excitation is somehow increased. So it tips this balance between excitation and inhibition. And that leads to difficulties in signal processing. So another set of studies have looked at the structure of the brain and the brain cells. And the brain cells are called neurons, and that's what they look like under a high-powered microscope. They have this so-called the cell body, and then all these branches, which are called dendrites, and then axons, which go to other brain cells, which we don't see here. But these little tiny protrusions on these dendrites, that's where the synapses are. So these are brain cells from a mouse brain, but these are brain cells from a human patient. And when people looked at the structure of these brain cells, they found that in autism, there are many more of these so-called dendritic spines. So look at this neuron versus this one. So that led to the idea that maybe there are also more connections in the brains of children with autism, which could explain some of the restricted interests, some of the repetitive behaviors. So this idea led to a model of so-called hyperconnectivity. So the brain cells in patients with autism are hyperconnected. So when you put the two together, sorry for the quality of this image, but in children with autism, we find that we have a combination of these two. We have hyperconnectivity, where there are too many synapses, too many connections between the brain cells, but also an increased excitation over inhibition. So these are thought to be very important contributors to the neurobiology of autism. So how do we study this? And as I said, genetics had a very important contribution, and these genetic studies have been done primarily over the past maybe really like 10 to 12 years. And what made it possible is that there were these very large consortia that included 50 to 100,000 patients and non-disease controls to pick up some of these genetic signals, to identify some of these genes, because some of these mutations are so rare, and some of the mutations have a small effect size. So to actually statistically analyze the difference between control groups and the patients, we needed this very, very large number. And also genetic sequencing technology is becoming cheaper. So, to sequence the human genome 20 years ago, it cost a few $1 billion. Now to sequence one person's genome, as I mentioned, is $1,000. So sequencing technology has become more affordable, and because of this, these large studies are also possible. And there are several of these ongoing studies. The Simons Foundation sponsors genetic studies. there are this Autism Genetic Research Exchange program, the Autism Genome Project, the Missing Project. So all of these have a slightly different approach, but they're all leading to new and new discovered genes. So they address this 80 to 85% still unknown genetic component. So why are these genes helpful? And I alluded to this a couple of times before. So the strategy would be like this. Basically, we do genetic studies on humans. We identify the genes that, when mutated, could lead to autism. And then we make these mutations in mice. And mice are easy to raise. You know, they start having babies like at the age of 2 months old. They can make many generations in a year, many babies. So we can do a lot of studies on them. And then using these mouse studies, which are called preclinical studies, doing these mouse studies can lead to drug targets, drug candidates, and help develop therapeutics. And then these therapeutics will then be used in patients. So that's sort of the outline of what I'm going to talk about today as well. But that is the general strategy of how, you know, basic science is used to understand the causes and then to find treatments for autism. So mice are cute little creatures, and we study them in different ways. And this is, unfortunately, this video for some reason doesn't work. But what we had here was, these are plastic cages in which we put a mouse, and then we have two little cages within this cage. And one has a new mouse, and one has it's empty. So a normal mouse would be very curious of this new mouse. And what this video would show is that this mouse, the control mouse, goes to the new mouse and starts hovering around, walking around this cage. So if you look at a mouse with a mutation in an autism-related gene, they have social behavior difficulties. So this mouse would walk and spend an equal time between this empty cage and the social cage, as if the mouse in the social cage, the new mouse, would not matter to the mutant mouse. Another way to look at communication behaviors in mice is look at so-called ultrasonic vocalizations. So this is an example of that, how mice sing to each other. For example, male mice use this type of song to court female mice, or baby mice use a different type of song to call their mothers. And mice have different types of these ultrasonic vocalizations. And this is just something. It sounds like almost like a bird song. This has been amplified because it's not in auditory range. It's not in the range that we can hear. That's why it's ultrasonic. But mice with mutations in autism-related genes have different types of alterations in this in these ultrasonic communications. Another way to study mice is by imaging. So this is something we cannot do in humans, but with mutant mice, we can attach a microscope camera to their head and look at these dendrites and dendritic spines that I mentioned before. So we can see if a genetic mutation leads to fewer or more of these spines. We can look at the activity of neurons. And here, this is a recording of the activity of neurons in the brain, and you can see these oscillations that neurons undergo. So that's what happens in our brains as we speak now, and it looks like mutations in genes that are related to autism alter these type of neuronal communications. So using mice, we can study all this. Another way to study autism in an experimental system is to study autism in a dish. So this is a new technology. The Nobel Prize was given in physiology a year or two ago for discovering this. Actually, this was a little bit. It was about 5 years ago, and it consists of taking blood cells or skin cells from the patient and reprogramming them into so-called induced pluripotent stem cells, which can be grown in a dish, and then these pluripotent stem cells can be differentiated, made into neuronal cells or cardiac cells or intestinal cells. So, but these would be coming from a specific patient and would carry the mutation that is present in the patient, but also all the other genetic information. So these would be a form of very personalized, almost like replica brain cells coming from a patient that are grown in a dish. So these type of brain cells, we will work with them in the lab also, and can be used in these high throughput screening approaches, where we put these brain cells coming with a specific mutation coming from a specific patient into these so-called multi-well plates, where and then we can put them in this image scanner, and we can put different drugs, or we can do different type of manipulations, and this robotic instrument can image them at the same time at very high speed. And then we get this type of information about the size of the brain cells, about the currents that they send to each other. So by doing this, we can test drugs very quickly, with not even in mice, but drugs on practically brain cells that are supposed to replicate the patient's brain cells. So this type of knowledge leads to a sort of a very different approach of understanding and treating autism in the future. So the classical approach, which is in practice now still in most of the cases, is when we expect to find one cure for autism, or one treatment that it's kind of a one treatment fits all type of approach for autism. And this is a slide from studies in cancer, where these type of studies have been inspired from. Now we look at, a patient is diagnosed with autism, and if they have the core symptoms of autism that I mentioned, and then we're looking for treatments. But these patients can be classified actually in subgroups. So, and they may respond differently to different drugs, or they may have different genetic mutations. So another approach for the future is a more personalized approach, where we group patients, we don't lump them all together, but we group them either based on genetic subgroups. So we study and we try to treat patients with SHANK3, for example, mutations, or CAPNAP mutations. And their treatment might be different than patients with 16p duplication mutations. And or patients with epilepsy and autism could be different from patients with autism only and no epilepsy. So these ways of subgrouping patients could actually lead to better treatments that respond much better, that are more efficient for one subgroup. But then when you mix all these subgroups and you give the same drug, the signal is actually lost, is diluted out just because of this variability. So the approach would be, in that case, study a larger population of patients, or do genetic testing, identify the genetic mutation, and then group patients based on these either genetic mutations or so-called deep phenotyping. And then their mutations can be replicated in mice, or these stem cells, iPS cells, can be derived from that specific group of patients. And then we can screen molecules, we can study them in mice. So we can do a lot of this biological studies and drug development here. And then we go back and develop specific drugs for these subgroups patients, or individually or in combination. So this is the approach that we are taking at the Center for Autism. And I should point out, and I also mentioned at the beginning of the talk, that we are primarily focused on this middle stage here. So we ourselves don't do genetic studies. That's done by others at a larger scale. We just take that genetic information, but we do make mouse models and we do make iPS cells, and we do screen drugs on them and find ways to treat these. And then the next step would be to develop these into treatments, which would be yet another complicated process. So at the Center for Autism and Neurodevelopment at Northwestern, we're focused on understanding the function of genes that are linked to autism and how this leads to disease. And this is sort of a general approach. I'm not going to go in detail about it, but I want to discuss 3 of our studies just briefly. So they start from genes that have been implicated in autism, and these sort of syndromic forms, these subgroups of autism. So each of these type of conditions you can envision as being one of these subgroups. So one of these subgroups is called Casper2 or CNTNAP2 deficiency syndrome. And this is caused by mutations in the gene called CNTNAP2. And this is just a cartoon of different mutations that have been reported in this gene. And there's a lot of heterogeneity even within this, because some of these mutations lead to autism, some lead to intellectual disability, some of these, many of them lead to epilepsy, and often they are comorbid. So patients with mutations in this, almost without exception, have seizures, most of them, and speech delay. So, and this is a paper from another lab that described this syndrome. So what we found, excuse me, was that part of this gene, or this gene makes a protein, and that protein is a very important protein at synapses. But part of it breaks off, and we can detect it in the cerebrospinal fluid of patients with autism. And these are actually real human patients that donated cerebrospinal fluid. And we can see a reduction in this, a portion of this molecule. So it could be used as a biomarker for patients that could be responsive to some of these treatments that we're developing. So next, we ask the question, well, can we just put this small portion of the protein back? So instead of doing gene therapy, in which we want to re-express the whole protein, in which people re-express the whole protein, and then they use viruses to infect brain cells, which could be quite risky. What we wanted to do is to just make this protein, which can be purified in a dish, and inject it kind of like insulin. So we tested this, injecting it first in mice, in the brain actually, directly in mice. And then and what we did, we replaced this protein in the mice that had a so-called knockout in the gene of catnap, CNTNAP2. So this is a behavioral test that's testing for working memory. It's called the Y-maze test because it's a maze that mice run in. And this color actually is a color coding of the position of the mouse. So usually you would put the mouse here and let it run. And the normal mouse would just go one arm, the other arm would alternate arms. But if the mouse is either doesn't remember which arm it went to, would keep going in the same arm. And that's what these catnap mice do. Or they have behavioral rigidity. They actually almost like have this compulsion to keep going in the same arm. So when we inject these mice, the catnap knockout mice, the CNTNAP2 knockout mice, with the CNTNAP2 protein fragment, it normalizes their behavior. and that's shown by this graph here. So that is a very interesting new approach in which is an alternative to gene therapy, because this way we can inject only a fragment of the protein and hope to rescue some of the functions. Another one of these genetic disorders is the Phelan McDermid syndrome, which is caused most often by mutations in the gene called SHANK3, which is a very important synaptic protein. And these lines just show the mutations of reported in different patients. That's sort of the clinical profile of these kids. So what we did here, we made a peptide based on IGF-1, which is an insulin growth factor, no, on IGF-BP2, an insulin growth factor receptor binding protein 2. And this small molecule we injected in mice. And also we tested these mice in several of these tests. And we tested muscle strength, for example, because muscle tone, because kids with Phelan McDermid syndrome have low muscle tone, and injection of this peptide rescued partially low muscle tone. We did the Y maze again, and it was a very good rescue, even improved the function to somewhat. And then we tested these ultrasonic vocalizations that I discussed a little bit earlier, which is a way to assess communication behavior in mice, and we could rescue this as well. So the third and last example I'm going to give is a chromosomal disorder called the 16p duplication disorder, in which one fragment of the 16p16 chromosome is duplicated. So this leads to a syndrome that is often associated with autism, about 20 or 30% of the cases, 30% of the cases with intellectual disability, seizures, microcephaly, speech delay, and so on. So what we found here was that we can inject another peptide into, this time again, into their brain, and we could rescue two of these behavioral phenotypes, behaviors that they show, specifically sociability and learning and memory. So basically that was my talk, and I just wanted to bring up a couple of resources here. So Autism Speaks is a great resource. They offer guidance and information about autism. The Simons Foundation for Autism Research invests a lot of money, and specifically in the science and in genetics. The NIH, National Institute of Mental Health, has information about autism. And then there's a site where all ongoing clinical trials are listed. It's called clinicaltrials.gov, where one could go and look for specific clinical trials that are ongoing now or that are recruiting, that recruit kids with autism in general, or with specific genetic mutations like Fragile X or Phelan McDermid syndrome or others. And then the Simons Foundation, again, releases this newsletter that showcases the latest research. So thank you very much. And this is my email. If you need to follow up with any questions or want to keep in touch, please email me. And yes, so sorry I ran over time a little bit, but please feel free to ask me any questions. I'm going to stop sharing. Okay. Yeah, so I see here there are a couple of questions.
Awesome. What a wonderful presentation. It was great. Yeah.
Well, thank you. So yeah, if you have any, there are some questions here in the chat box, but also if you have any questions, just please turn on your camera and go ahead and ask.
You had mentioned earlier that trisomy 21, is that relate? And I lost a little bit of what you were saying, but is that related to autism, or is that something that if someone has a fetus that has trisomy 21 tested genetically for it? I mean, like, this is very new to me. Someone who had that beforehand and now has a child with autism, and no one ever said anything about, oh, there could be a relationship between the two.
Yeah, so the trisomy 21 I brought up more as an example of a type of chromosomal disorder where you have an entire chromosome triplicated, basically. So some kids with trisomy 21 do have autism. So, you know, autism, if you look at it from the perspective of the symptoms, looks one way, but if you look at it from the perspective of genetic mutations, it's a very different condition. So you have, you know, many different diseases caused by genetic mutations, many of these syndromes, and kids or carriers of some of these mutations have autism as some of the clinical presentations. So, you know, it's almost like autism is a phenotype or a clinical presentation of kids with different genetic mutations, as opposed to being an entity in itself. So I don't know if I'm clear about that, but in that sense, you know, various types of genetic mutations, including trisomy 21, could lead to autism. But in trisomy 21, I think that the frequency of autism is much lower. Yes. Wendy?
Yes, thank you. I did send a couple of questions, but I'll go ahead and ask them. We had my daughter's genes checked about 20 years ago, and it sounds from your talk like it might be worth checking again. They didn't find anything notable 20 years ago, but it certainly does sound like there have been many updates since then. One thing that has come across my desk is Spark, and I noticed one of your slides had a Spark emblem on it. Do you know anything about Spark, and is that a good way to perhaps get my daughter's genetic testing done?
So yes, so Spark is free. You can participate in Spark, and I can send Tia a link. And there's one site here. There are many intake sites in the country. So it's free, and they do the genetic testing, but you only get it 2 years later.
That's okay.
That's kind of the drawback.
That's okay. I didn't realize until I looked deeper into Spark that it is reputable. I was not aware. It seems to have maybe 20, maybe 30 different collegiate allegiances associated with it. So, so it's good to see that it is a way to go.
Yeah, so there is one way, and it's free, and they do the interpretation. So, you know, it's not just the test, but also the genetic counselor, in a way, is included. But it does take a while.
You do get individualized information. It's not just made part of a database and only they see it.
So it's anonymous to everybody else, but they send you the information as a donor.
Oh, okay, that sounds great.
Yeah, and so, you know, the information is used in, you know, these genetic studies, but it's anonymized. So that's used immediately, but you get the information only 2 years later.
Okay, that sounds wonderful. This was the best talk on this topic that I have ever seen.
Oh, thank you.
I really thank you for this. I really feel like I have a much better understanding of what's going on. So thank you.
Well, thank you very much. Any other questions?
What do you mean? Yes, I have a question. Our daughter had genetic testing, and it was identified, it was epilepsy testing. And it was identified that she has some mutation in SCM8A gene, sodium channel.
Yes.
We didn't do autism testing, but epilepsy we did. And she has occasional seizures, some autism, and some intellectual disability. So we do know that probably the cause, but the question is, like, how far research from implementation, like what steps we can do now if you identify the mice and the treatment, how can we actually get the treatment?
Yeah, so the first thing would be to reach out, and this is available in, you can probably do a Google search to see if any labs or any scientists study specifically that mutation. And they can guide you to clinicians who see kids with that mutation. So for example, here in our center, there are several labs that study other mutations, SCN1A and SCN2A genes, so related, but slightly different. So they're also working with clinicians at the pediatric neurology at Northwestern, and they can put you in touch with ongoing clinical trials, if there are any, or so that's one way to just track down the labs, and that's from a Google search. And
I do know the trials, but she doesn't fit to any trials because she doesn't have seizures often enough. So what you mentioned by autism, she might fit to trials.
Yeah. So yeah, exactly. So I think that there are current